I'm going to talk about biosphere complexity as a possible new approach for defining life. So first of all, what is life? When we see life in all its brilliant different kinds that we notice in nature, and they are all so different that it sometimes feels hard to imagine that there is a common concept that you can really easily grasp. And that's what I did. I just put some of the existing definitions that are published all in Wordle, and let it calculate this here. And then you see what is the typical wording that comes up when you talk about definitions of life. Of course, definition of life, but you also see major things like evolution, Darwinian, its systems seems to be a huge thing. Then information is and chemical is maybe some of the largest too, and et al. Obviously for the citations. When we talk about definitions of life, I think we have to look at the different semantic meanings that are behind the term life. The term life is very heterogeneous. They used in language, so there are different ways of thinking about life. Life as the concept of dead versus alive, or the non-living versus the living matter. Life might also be just a transitional quality like shelf life or half-life, which doesn't even necessarily have to do anything with biological life, right? Maybe a drug has a half-life or a shelf life. With artificial systems, if we want to know if a computer should be called alive, then we might put different measures on that, as if we want to know if a human person is dead or alive, or if we are in the stem cell debate or birth control debates where we want to know where is the beginning of human life. These are completely different questions, and so to my mind they all have different aims, address different questions, and need different solutions. So in this way, we won't find one definition of life that fits all of them. So we need to focus on what we're interested in. And in this case, I want to talk about life in the context of origins of life and quest for life in the universe. And if we want to define that, there are a couple of requirements for such a definition that I think are the most important. And in general, I would think such a definition about of life in the context of its origins and the search for life in the universe needs to be universal in time and space. It needs to fit with our current picture of physics, chemistry, and biology. And it must include life on Earth, but should be also open to other possible, thinkable chemistries or whatever we might encounter out there. So it shouldn't be not too specific that we say it's like you need a DNA strand or something, because it's most likely not what you would expect to find exactly as a copy of life on Earth. Then it should be useful, so of somehow operational value, and help to identify life in the universe. So if you find something somewhere else, can you use this definition to infer if this is life or not? And it also should make, to my mind, testable predictions and allow to generate hypotheses on the origins of life. Now let's look on two existing definitions. One, the standard definition that you might all know, the NASA's working definition, life as a self-sustaining system capable of Darwinian evolution. Another one autopoiesis, which I just put in a really very stripped down way. There's more to that, I guess, than this sentence, a system capable of reproducing and maintaining itself. But if we look at both of them, we always see one term, which is like maintenance, self-sustaining, and one system of reproducing or evolving. And if we look at virtually any definition of life, we'll find it comes all usually down to three things. There is the unit, which is a system, a membrane-bound structure, an organizational form, a confined reaction room, barrier-enclosed structure, whatever you might call it. Then, second, the capability of evolution or reproduction, which in some way is synonymous, because when you can reproduce and you can reproduce with and produce mistakes during reproduction, then you might also have the ability to evolve, and there fall in all different wordings and phrases that are typically used, from Darwinian selection, adaptation, template, code. That's all more or less directly or indirectly referring to evolution and reproduction. And third, we have metabolism terms in the definition, which usually refers to some degree of autonomy, self-sustaining, maintenance, self-repair, energy, conversion processes, production of its own constituents, transformations, chemical self-construction. So if we break it up, finally you can always find somewhere these three incorporated in it, and I would think if you just pick out any word of the three, or at least any one word of the three, then you can always make up your own definition of life, which sounds some way like something that most people would accept, maybe in some way. So you could say it's a membrane-bound structure capable of reproduction that is self-sustaining or something. Yeah, that doesn't sound too bad and fits maybe roughly with what we are have in mind about life. So, but let's go step by step. So, what's the unit of life? And here I will argue that it's kind of blurry, everything. So if we really look at what kind of units we see in our current life in biology, then we'll find we have, if we focus on cellular architecture, then we have organelles, and some cells do have these organelles, and we would see the organelle as the subunit of a larger thing, which is the cell. And in another case, we would have maybe a whole organism, which we see as unit, and the cell as the subunit of the organism. So there we already have some kind of problems of defining what is actually the unit. If we want to talk about the unit as the unit of life. So what of these is the actual unit that you would call life? Is an organelle maybe a life? Is it a cell, or can it only be a higher or an organism that's like an individual unit? And if we look at organelles, for instance, then you'll see organelles, of course, we know today are derived from endosymbiosis. So they originally came in as an independent bacterial-like organism into the cell and are taken up and integrated now as part of eukaryotic cells. And if we plot the genome size of organelles and small endosymbiotic bacteria against the number of encoded proteins, then you will see that we actually have a really gradual transition. So here are maybe that's all organelles, the circles without filling. So you can see some mitochondria there. You would clearly say, okay, that is maybe not the unit of life if you have the mitochondrium of Plasmodium falciparum that has maybe 3 functional proteins encoded by maybe a number of, 8,000 genes or something that's in this organism or in this organelle. But if you then go up to some larger things like chromatophores or the plastids of plants, then you will see they have an amazing number actually of functional proteins encoded in their genomes in these plastids. So the plastids have a lot of stuff they are making themselves, and they are pretty large. And actually they are as large as some of the smallest known parasites and endosymbionts. So there are a lot of symbionts that are actually a little bit smaller in genome size and have less functional proteins encoded than some chloroplasts. And we have, for instance, this Paulinella chromatophora, which is a plastide, which is actually much larger and contains much more functional proteins than even Mycoplasma, which is a pathogenic organism that infects humans. And I guess no one would argue that Mycoplasma or chlamydia is a part of us. It's rather something else that infects people. So it's in this case, I think this shows the great continuum between unit and subunit. So in one case, you will argue that this is clearly a subunit of the organelle is a subunit of the malaria parasite. But on the other hand, with some of these endosymbiotic or parasitic organisms, you will never see them as a subunit of us or any higher organism. But still they are in a kind of a continuum. Then when we look at a larger scale, then we'll see that there's also the borders blurred between the individual cell as the unit of, as a subunit of a higher organism, and the organism itself as the unit of selection. So if we look at this is the Tasmanian devil, and these are really aggressive animals, so they usually fight and bite each other in the face. And there has one cancer cell line developed from a female Tasmanian devil. And this cancer cell line has developed to a transmissible parasite. So that's why this species has now a huge problem. A lot of them are dying because of these transmissible cancers. And just if you imagine that a cancer cell line, which emerged from a somatic cell, which was actually only a part of the organism and is usually would usually die with the end of the life of the organism, does in this case not die, but becomes its own, becomes a parasite of the species and has some, is the unit of selection basically, and not anymore a subunit of the organism, and can be transmitted from one individual to the next. So I think this shows also that we have here a really blurred boundary between unit and subunits. And finally, if we really look at the boundary, and that's what in autopoiesis or autonomous systems is usually claimed, that it's something from within, that we have a cell as a boundary structure, and everything is created inside the cell. The cell self creates it from within or something. But I mean, this is our typical picture. if we look at cells, then we typically have in mind, okay, it's a enclosed boundary structure, and everything else is outside. It takes up nutrients and pumps out the waste, and everything else it does inside the cell. But actually, if we really look at cells, how we should see them, actually, it is much less clear what is actually where is actually the boundary. If we come closer and closer to the cells, we will see a lot of membrane vesicles that are budding and that are fusing again and exchanging compounds and particles. We have a huge network of extracellular material which cells adhere to, and a lot of cells actually, for instance, the most cells of our body, they will kill themselves if they don't find themselves attached to this extracellular matrix. So is this extracellular matrix then a part of the unit, or is it does it belong to it? Then a lot of metabolism is actually not taking place in the cell, but outside of the cell. So we have nanovirus in some organisms that are doing redox reactions outside. We have enzymes, extracellular enzymes that are doing reactions outside of the cell. Even on the flagella, it has been shown that there are ATP synthases running along and producing ATP outside of the cell on the flagella to supply them instantaneously with fresh ATP. We have pili, we have membrane proteins, even the cell wall, even the construction of the cell wall mainly takes place outside of the cell. So there are enzymes linked to the cell wall, peptides that connect them again and make them larger. So a huge fraction of the machinery of the cell actually takes place outside of this strictly defined boundary, which is not that strict at all. Okay, let's look at metabolism and replication if we I go a bit faster through that. So there are units that you would say are units of metabolism and units of evolution, and that's maybe more the traditional view that you would adopt. Okay, there are some that can only evolve and others can only do metabolism, and you have some that can do both, and that's mostly what people call life. But if we look closer, then we'll also see that there is a highly gradual and conditional capability to evolve. A red blood cell doesn't have any genes anymore, so there's no capability to evolve. An asexual organism has a full capability, so it just needs to divide. But we have everything in between, so sexual organisms depend on a second partner, so it's more conditional than an asexual organism. A spore or a seed will first find the right conditions to germinate, and then it might reproduce via sexual reproduction. So it has more conditions on it to be capable to evolve. Then finally, we have some sterile organisms, social worker castes. They have still cells in them, so you could take them out, immortalize them, and have a cell line from these organisms, as you could do maybe with an endling, which is the last species, the last individual of a whole species on the verge of extinction, so like the last Tasmanian wolf that then died. And so there you don't have any capability of evolution in a direct way, but you still could, of course, by artificial methods or by taking out some cells, gain maybe some degree of evolution. Naked DNA can be taken up by a cell, integrated, and then is also capable of further evolving. So there is more and more a gradual and continuous thing. And if we look at metabolism, we'll see the same thing. There are bacteria which have basically everything they need by themselves, but not every organism does produce and metabolize everything by itself. So we have parasites, of course, that need a lot of metabolic activity from outside. We have red blood cells which cannot do anything everything by themselves anymore and need a lot of input from outside. And we finally come down to the lower end where we have enzymes or a virus that can only maybe just hydrolyze one ATP molecule and inject itself in the cell. So if you want so, then this one hydrolysis step is also a metabolic step, but at a very, very low level of capability of metabolism. We see these limitations everywhere. So we have chlamydia that can not synthesize their own ATP, so they cannot even generate their own energy currency. Borrelia, another parasite, cannot even synthesize fatty acids, so they are not even capable of making their own membranes just by their own metabolism anymore. And even if you look at us, we cannot produce everything that we actually need. And so what is then life? And I would just shortly come to that. Let's look at what is the common ground of life. We need structure for metabolism and function. And this structure requires molecular complexity. And on the other hand, we need somehow information for replication and evolution. And that I would term informational complexity. So we have two types of general systems that we need combined so that metabolism and evolution can happen, to which degree. And so I'll define this as the biogenic unit, any entity that comprises both molecular complexity and information complexity. And looking now at what actually molecular complexity is, I derived an equation that is very broadly applicable and that you can use for very small molecules to large molecules, and that gives you mathematical value, a fixed value for a molecule for its molecular complexity, and that was just recently published, and I just shortly flip through, so you will see that the small molecules have less complexity, larger molecules with more substituents, with maybe more heteroatoms or more stereochemistry, they have higher complexity, and that also typically fits what we have in mind if you look at this chart of prebiotic compounds. The complexity increases with increasing molecular structural complexity. And the second type of complexity, that's the information complexity, but that's easy to calculate. That's just as you do it with DNA or any other code by the Shannon formula. So finally, we can map information complexity against molecular complexity and find out that we get this complexity space map where we see how units, how these biogenic units behave in this complexity space. And we see that we get small peptides, enzymes, we can calculate exact values for everything. And we can also go to the larger units and just look at these are whole proteins. But you can also calculate a whole virus particle, which is very well defined in case of the tobacco migraine mosaic virus. So you can calculate for all proteins, for the DNA, for everything that's contained in the virus, the complexity, add that together, and you'll end up here at this value. And you can do the same for genomes, but of course it will be harder for whole organisms and cells because they are now rather statistically composed and not anymore by a fixed value of how many proteins an E. coli cell has. So you have rather to you have to approximate the number of proteins that are in the cell and of lipids and so on, and then you can calculate actually also complexity for larger units in the biosphere. So you can do that for very small things still, like viroids, or that's the Taq polymerase, for a whole virus. For E. coli, a HeLa cell, that's the cell of the baker's of the yeast for beer brewing. That's of a nematode, a small worm, And if you calculate it for the number of cells that a human has, then you end up at Homo sapiens. You can also add up the microbiome of Homo sapiens, then you get to this complexity value. Finally, if you add all interacting humans in the world, then you end up with mankind, and you can even add the information content that is in mimetic evolution in mankind, which is, for instance, the whole written record of mankind, which I just roughly calculated what this information complexity would be, and so you end up with a whole map of complexity space of the biosphere. And so finally, we can say, what is life? I would argue it's not the unit on the individual scale, but we must see life as a phenomenon of a whole biosphere. That means life is the property of a biosphere as a whole system that acquired the necessary level of complexity to be capable of evolution, and that depends on the evolution of a template-directed replication and information transfer strategy, which is, for instance, given by DNA or RNA, and you can directly see this in these charts where we stand here. I don't argue that you can now exactly define on the point life begins here or something, but you can say where does the system range, and so you can, in the whole gradual space of what might exist out there or what might have existed in a prebiotic world, map where things are, and even come up maybe with ideas, how do we get from no complexity, or if you have amino acids, they would range around here, how do we get to something as complex as maybe the tag polymerase, and what trajectories would we have to take in this complexity space? Do we have to first increase maybe in molecular complexity and then gain a little bit of information complexity, or can we even go the direct way, for instance, by building up peptide chains? And the good thing is the whole thing is completely adaptable to any complexity of even non-carbon chemistry, so you could use that for even boron chemistry, silicon chemistry, whatever comes to your mind, and how plausible or implausible it might be. And so that's the final slide. The conclusion, I won't read that anymore to you. I thank my funding agencies and shortly a view of Constance. If you have ever wondered where it is between in Germany, but at the border to Switzerland, at this beautiful lake, and come to visit. Thank you.